Hey guys, welcome back to a new video by Biology with Zhangxuan. So today we are going to attempt the Cambridge IGCSE Biology Paper 4 Theory Extended to October-November 2022, Paper 4 Variant 2. So if there are any questions, do comment in the section below as we walk through the entire questions and answer in depth together with explanations. So let's start with it. Question 1. Phytoplankton consists of many species of single-cell and many-cell algae. So single-cell is unicellular, many-cell is multicellular. So part A, algae are classified in the Prototist kingdom. All algae contains one or more chloroplasts. State the name of another kingdom that contains organisms which have chloroplasts. So chloroplast is of course the pigment. Okay, it contains the pigment which is the chlorophyll that gives that green appearance on the organs of a plant. And the most obvious one would be plants. A student investigated the species composition of the phytoplankton in a lake. Figure 1.1 shows some of the phytoplankton collected by the student. So part B, the actual length of the alga X is 0.19 millimeters. So this is in millimeters. And now they want you to convert the actual length to micrometers. So micrometers is in μm. So 1 mm is equals to 1000 micrometers. And 0.19 mm, you just times by 1000, you're going to get 190 micrometers. So this is the answer. C. The student made careful drawings of 7 types of algae found in the samples of a lake water. The drawings are shown in figure 1.2. So they've given you 7 specimens. And now they ask you to do a dichotomous key or dichotomous key. So you use the key to identify each of the species A to G. Write the letters of each species in the correct box beside the key. So this is the full answer. I wouldn't be able to explain this. This should be based on your observation. But if you have any questions, do comment or say why does this is answer G, why is the answer D, something like this. Okay. Part D. Phytoplankton are photosynthetic organisms, means they can undergo the photosynthesis. So describe the importance of phytoplankton in the food web of the lake ecosystem. So we know that they are photosynthetic organisms. It means that they are considered as a producer in the food chain. They help to produce the food for the herbivores, okay, for the primary consumers and also the rest of the food chain. So what is their importance? Okay, just describe their importance. So first thing, you highlight that it's a producer. The moment you highlight a producer, you link is that it makes food from the sun because it undergoes the photosynthesis. Okay, and what you do is that after they absorb the light energy, they will convert it to the chemical energy. So from there, they will start to make their own food. Then this helps to provide, you show what is role of the producer. The role of the producer is to provide the food for the second trophic level, or you can say the primary consumer, and energy throughout the trophic level, because producers have the highest amount of energy and it's also very useful to be transferred. Okay, it has the most energy. Okay. Question 2. Figure 2.1 is a diagram of the human alimentary canal and associated organs. So they provided you all in numbers and they should be asking you for to fill in something. So part A, table 2.1 shows organs that secrete these enzymes, okay, and their substrates and the products. Okay, so this is 4 marks, meaning this whole column is 1 mark. The second column, 2 marks. Another 1 more mark again. The third column, 1 more mark again. And the last column, 1 more mark again. So the total will be 4. So meaning if you get one of these incorrect, the whole column is 0. Okay. So amylase, it's the organ that secretes is the salivary glands. and it's located at number 1. And the substrate, meaning is like this is the enzyme, okay, amylase is the enzyme. So what attaches to it so that it catalyzes the reaction to form something else. So this is the starch, okay, so starch, okay. So the starch will be broken down to form maltose, okay. Then you have pepsin. Pepsin is located in the stomach, and the substrate with the protein, and they will form the amino acids, okay. And the lipase is secreted from the pancreas. The substrate with the fat, they will break the fat down to fatty acid and glycerol. Okay, be careful, glycerol, not glycogen. For the last column, maltase, is located in the small intestine. So what we do is that they use the maltose and break down into glucose, to into a smaller one, and they ask you to identify which is the small intestine is located at 5. Part B, explain the role of hydrochloric acid in the alimentary canal. So, the hydrochloric acid is located in the stomach, okay? And what they do is that they kill the bacteria. So that's why our hydrochloric acid is so useful. If we, of course, food itself may have bacteria. If it's contaminated, for example, there's some potential for the hydrochloric acid to, to basically kill everything, and they will denature the enzyme in the bacteria. So because bacteria are also a type of living organism that may have enzymes that are part of inside them. So what happens is that because of the acidity of the hydrochloric acid, they are able to denature them, okay, and it's a suitable pH for the enzyme pepsin to work efficiently. Part C, state the function of the region of the alimentary canal labeled 6 in figure 2.1. So it's to reabsorb the water or ions, or can be salts, okay? So there's no digestion or anything in the large intestine. Part D, describe how food is eaten by humans is reduced to smaller pieces, okay, and explain, okay, they ask you to explain how this makes chemical digestion more efficiently. So this is 6 marks. We try to break it down. Let's see how can we describe the process. So since they say to make chemical digestion more efficient, it has to undergo one process first, which is the mechanical digestion. So this is the major keyword. Your answer must be linked with a surrounding this word, and it's by the mouth or the teeth. So mechanical digestion will be done firstly in hand before the chemical digestion occurs by chewing, okay? So with our teeth, okay, you can see the teeth, like in scissors, you can state a few of our teeth that helps to cut food, and the molars that can grind the food, okay? And what it does for making chemical digestion more efficient is by increasing the surface area of food so that the enzymes can act easily on it. Okay, so enzymes can act easily. Okay, then you move to the stomach. The stomach, okay, there's a churning. Churning, it means that because the stomach has a lot of layers, okay, so stomach looks something like this. Not a very good drawing, but it has a lot of layers here. So churning is like, you can hear when you're hungry, right, you start to hear those goo goo goo, that type of sound. So that's churning. So this is a part of a mechanical digestion that occurs in the stomach, that which helps to to move the food and to form a chyme. So chyme is the liquid form after digestion in the stomach. Okay, you go back to this diagram. Okay, you can see when food enters, okay, it will be undergoing the mechanical digestion, right? So after it has formed it finishes digestion and everything, it starts to form a kind of like a globule-like structure, and this is what we call as a bolus. Okay, so after the bolus enters the stomach, okay, it will undergo this churning and also the hydrochloric acid. Then they will start to form some liquid in there. So the liquid is called the chyme. So it becomes that liquid form, which is much easier for absorption later on. So that is what is called as a chyme. Okay, that's the small intestine. It allows the emulsification of fats by bile. So bile, we all thought is chemical digestion, but it's not. Chemical digestion must be involved with enzymes. Bile is not an enzyme. Bile is just it's an alkaline liquid that helps to emulsify the fats from larger droplets to smaller droplets of fat. It's not from fats to fatty acid and glycerol. It's different because it's just breaking down that so big molecules of the fat into a smaller one. Okay, so that's how you get your 6 marks. Question 3. Aphids are insects that feed on the phloem sac by inserting their mouth parts into the phloem tissue. Figure 3.1 shows an aphid feeding on phloem tissue in a leaf. So A part 1, state 2 features visible in figure 3.1 that identify this animal as an insect. So this comes back to chapter 1. Okay, so how do we identify this animal as an insect? You just see visible in figure 3.1, there are 6 legs. Okay, and you can see that there are 3 body sections. 3 body sections, so you can also state that that's a feature also. Part 2, aphids can transmit viral pathogens, okay, when they feed on the plant. Suggest how severe infestation of aphids on crop plants can be avoided. Okay, so to prevent that many of the aphids are present in that particular location, you can use insecticides or pesticides that which are able to inhibit the growth, the severe infestation of that aphids on the crop plants. Or maybe if you don't want to use insecticide or pesticides, you can move your crops into glass houses which have better protection from the environment, and you're able to control the limiting factors. You can control the condition of how it's able to grow inside the glass house. And another thing is that you can avoid monoculture, because monoculture is only one single species of particular plant that is grown in huge amount. When there's huge amount, the DNA is the same. So when the DNA is the same, they are very susceptible for disease to attack, and it can spread very quickly because they do not have a unique DNA. Like humans, we have different appearances, okay, of the same species. So sometimes it is not necessary that if you got disease, another person will get it also. So because of the difference in the DNA, okay. And part 3, the aphid shown in figure 3.1 is feeding on the phloem tissue in the leaf. State the name of the 2 tissues in the leaf that the mouth parts of the aphid will pass through to reach the phloem. So typically this is located in a leaf. So what happens in a leaf is that the xylem is on the top. Okay, so the xylem looks something like this usually. It's cut into half. So in order for it to really penetrate in, right? So this is the palisade mesophyll cell. So they say 2 tissues, okay, in the leaf. This is the mesophyll cell. So what happens is that the aphid, right? It will insert itself into the upper epidermis and then to the xylem. Then only it will reach to the phloem. So they will penetrate 2 tissues, which is the xylem and the upper epidermis. Okay. Part B. Aphids have been used to investigate the translocation of sucrose in phloem tissue. While they are feeding on the phloem sac, aphids excrete a sucrose-rich fluid known as honeydew. In an investigation, 2 groups of 4 aphids were placed at intervals along the stem of a young willow plant, as shown in figure 3.2. So you can see that they set up an investigation, and so the leaves were enclosed in airtight glass chamber. So you can see this is airtight. A special form of carbon dioxide gas that contained the radioactive carbon-14 was supplied for the leaves in a short period of time, and the samples of the honeydew were collected the time taken for sucrose containing the 14 carbon travel the distance between group A and B was recorded. So the investigation was repeated twice, okay, using a fresh stamp, okay, and the different groups of athletes for each trial. So the results are shown in table 3.1. So they asked you to complete the table 3.1. So you can see that the distance between group A and this, the time taken for this, you just do. Hold up, let me. So this is in minutes. Okay, so you can see this is in minutes. You check the unit. This is in millimeters, and the rate is in mm per hour. So you have to convert this 120 to hours. So 120 minutes is equals to 2 hours. Okay, we try to do the easier one first. Okay, so you can see that 340, okay, in mm, 75 is about hold up. So 1 hour 15 minutes maybe. Okay, or you can put this as or maybe you can put as 1.25 hours, which you just take 15 divided by 60. Let me try to. Yes, you get 272 and 630. You know that 2 hours is 120, be 2 point something. So 150 minus 120 will be 30 minutes. So 30 divided by 60, you're going to get half. So it's 2.5. So 630 divided by 2.5, okay, 2.5 hours, you're going to get 252 mm per hour. This is the answer. Part 2. Outline how carbon-14 in carbon dioxide gas becomes incorporated into the sucrose molecules that are translocated in the phloem. So how the carbon dioxide gas, okay, is shown as it's a gas, okay. So when you know it's a gas, it's entering into the sucrose molecule, it's like being part of it, it's definitely by diffusion. So first thing, I already know that it's going to be diffusion. And how is it going to enter from this into the stomata? So they'll enter the leaf by the stomata via diffusion. Okay, then they will consume to make the glucose because that's the main form it has to undergo. Okay, and then it only moves into the chloroplast of the mesophyll cell and being part of it with the sucrose. Part C. Sucrose travels from stem to other plants known as sinks and where it's used. State two parts of the plant that are sinks for sucrose. So sinks and source is about the same thing. So sinks, you can say, is a flower and a root. So they have different times and with different seasons, right? The sinks and source to interchange, okay, depending on the condition and the suitability of the environment. Part 2. Sucrose is used in the cells of sinks in a plant. Describe the uses of sucrose by sinks. So what they do is that in the sink, they will convert the sucrose into a more soluble form, which is glucose. And glucose is it's a viable molecule to undergo respiration because respiration always needs 6 carbons. Then they will provide the energy in the form of ATP, and when the ATP is produced or the energy is being supplied, they can be used for reproduction, active transport, and then once it's done, it's going to be stored back as sucrose again. Question 4a, figure 4.1 is the flow chart showing some of the processes that occurs in the biofuels power plant. So you just look at it, eventually it's going to link to an anaerobic respiration. So part 2, the fermentation stage shown in figure 4.1 requires yeast. Complete the balanced chemical equation to show how ethanol is produced by yeast respiration. So this undergoing this anaerobic respiration in the alcoholic fermentation. So you must know how to balance the equation, and you know this is the ethanol, 2 carbons. Okay, so it's basically, if you take chemistry, you know ethanol will just look like this. So you know there is and its ethanol is an OH there. Okay, so this is ethanol, and then the plus 2 CO2, then you just balance it accordingly. And it's basically just 2 and 2 only. So part 2, using the information in figure 4.1 suggests the environmental advantage of using ethanol as a fuel. So first thing, you must know that ethanol is originate from farming. When you farm, you're able to get from the plants this ethanol. Okay, and what happens is that they can reduce the non-renewable resource. Okay, so it's considered as a biofuel, which is a renewable source of energy, which we can decrease the reliance on non-renewable resources and therefore lesser carbon emission, and therefore there will be no contribution to the greenhouse effect by producing all the unwanted toxic gases. Okay. Part 3. Farmers grow giant reed plants as monocultures. So describe the disadvantages of growing giant reed plants to provide biomass for the production of biofuels. So first thing, they are used for food production. Secondly, there are loss of habitats and biodiversity, soil erosion, because the disadvantages of growing giant reed plants. Okay, part B. One problem with using biomass in the process shown in figure 4.1 that is the breakdown stage produces sugar called xylose and ethanoic acid. Yeast cannot use xylose and ethanoic acid is toxic to yeast. So scientists genetically engineered a type of yeast that can use xylose and ethanoic acid. So figure 4.2 shows the result of one of the trial experiments done by the scientists using their new genetically engineered yeast. Okay, so they've given you the graph. So part 1, describe the results shown in figure 4.2. So what happens is that in general, okay, this is described is you see the general trend of the curve. So you can see the concentration of the xylose, okay, this I'm referring to these two graphs, it has decreased. So it's going to a downward trend. However, you can see that the concentration of the ethanol is going up. It's an increasing trend. So the steep, it decreases until 62 hours for both xylose and ethanoic acid. They'll begin to stop their steepness, okay, at 62 hours. And the increase in ethanol concentration to up to 44, okay. So this is their about the midpoint. So to 44 gram per dm cube. So which is about here. 44 around here maybe, approximately 44. Okay, so that's it for the result shown. Part 2, the experiment was done at 30 degrees. The scientists repeat the experiment at 20 degrees. Predict the results that you would expect for the concentration of ethanol. There'll be a decreased rate because at lower temperature, what happens is that the rate of respiration is much lower also. So this is the answer. Slower rate of increase of ethanol, meaning that there will be a much lower, it takes a longer time to reach that particular point that it needs to reach. Okay, question 5. Figure 5.1 shows a McKinley cuckoo dove, so Macropigia mackinleyi, which is found on most of the islands in the southwest of the Pacific Ocean. Kaka Island is one of the islands where McKinley cuckoo doves is found. The species is part of many communities that are adapted to the different habitats on the island. So define the term community. So community is all of the population of different species in an ecosystem. So the keywords are all of the population of different species, okay, in an ecosystem. So you must memorize this definition, okay. Part 2, complete the passage with the most appropriate word or phrase. So adaptation is sometimes defined as the process resulting from natural selection. So natural selection is, of course, the best of the best that will survive and move on to the next coming generations, by which the population becomes more suited to their environment, or you can say surrounding, over many generations. So adaptation is, this is the definition. Part B, the highest point on the Kaka Island is 1,800 meters above sea level. In 1969, a researcher surveyed the bird species on the Kaka Island. He recorded the vertical distribution of the bird between sea level and 1,600 meters. In 2013, other researchers repeated the survey. So figure 5.2 shows the ranges of 4 species as recorded in the 2 surveys. The vertical lines represent the range of height where the birds were seen on the island. Okay, so you can see that there are different ranges between 1969 as shown in the more thicker line and 2013 in a more thinner line. So part 1 said to compare the range in 1969 with the range of in 2013 for each of the bird species, meaning they are referring to A, B, C, D. We're referring to all of these. Okay, and now showed in figure 5.2. So for A, you can see that the range goes downwards in 2013 and the altitude has also decreased. Okay, for B, it of course becomes a higher altitude in 2013, and for C, there's not so much change because it's still within the range. Okay, you can see that this too is still within the range, but for this, you can see that it has already go down, a decrease in the altitude. For this, you can see that it only is part of it here, but you can see in 2013 it's extended at a higher altitude. For C, that's of course I've shown you that there's not much change, and for D, I don't think there is also much change, no change in the ranges. For part 2, using the information in figure 5.2 suggest reasons for the change in the range of the island thrush on Kaka Island. So you can see that why is there a sudden decrease for the thrush. So why it could be a decrease in the population. And then you can see that there could be a possible competition for the food and the habitat that may be available to them. They could die, they could decrease in population maybe due to the disease, predation from predators, and could be from surrounding factors like habitat loss or deforestation. Part C. Small oceanic islands are often inhabited by species of bird that are found nowhere else. Many of these species have decreasing population and are often endangered. So explain the risks to these species of bird that have decreasing population. So what are the risks? So of course the risk is that it could be very hard to find a mate so that they could not reproduce anymore due to this. Inbreeding could be one factor, decrease in the genetic variation, and they can die due to inherited disease because there's a lack of the variation, and of course a higher risk of extinction. I would say that a lot of people would come to their first in mind is extinction. Okay, but this is the step by step. It's also acceptable. Question 6a, figure 6.1 shows diagrams of the circulatory system of a fish and a mammal. The arrow shows the direction of blood flow through the circulatory systems. So you can see they provided you the fish, which is a single circulatory system, and the mammal is a double circulatory system. All right. So part 1, they say state the names of the structured label Q. Okay, Q, T, and I think it's W and X. W and X. Okay, let's see what's Q. So Q, they're pointing you, this will be the heart. Okay, so heart would be unacceptable. I would say some may see as left atrium, which I would say yes, also works. T, so T is the kidneys. Okay, it's going out from the kidney and it's going back to the heart. So going back to a heart is a vein, and kidney is called as renal in medical terminology. So it's a renal vein. For W, you can see this is the major vein that goes back to the heart. So this is the vena cava. For X, you can see that it's going back to the lung, and it has the only artery that carries deoxygenated blood, which is the pulmonary artery. Okay, that's it. Part 2. State the name of the structured V on figure 6.1 and describe its function. So V is located in the center between the left and the right, which is the septum. The septum, very simple. They only separate the oxygenated and the deoxygenated blood. Part 3. Figure 6.1 shows that fish and mammals have different types of circulation. State why the fish circulation is called a single circulation. So I feel like you may be wondering what I mean by why. It's actually just a definition of single circulation. So definition of single circulation, which is blood that passes through the heart once. Okay, that's the key word. In a complete circulation, in one complete circulation, the blood only passes through once only. Question part 4. Explain the advantages of double circulation of the mammal, which is also a familiar past year question. Why is that it's an efficient supply of blood to the body, so that they can send blood in higher pressure, efficient removal of the carbon dioxide, decreased pressure in the pulmonary artery. So you see here the regulation of pressure can differ the location it's going to send the blood towards. Okay, and of course the lungs is very close to the heart. They do not need to send the blood at such high pressure like the arteries, and more time for gases exchange also. Okay, part B, the blood vessels that supply the heart muscle can become blocked. So part 1 says state the name of the blood vessel that supply the heart muscles is the coronary arteries to the heart muscle. So the heart muscle is also a muscle that will contract and relax and undergo respiration. So it is sent by the coronary arteries. Part 2, state one way in which blockages in these blood vessels can be treated. Angioplasty. There's a lot also. Stents are also acceptable. So there are many answers. Okay. Part C, substances move between blood and tissues at various sites in the circulatory system of mammals. Part 1, oxygen is absorbed into the blood and it passes through the lungs. Okay. And state the structure of in the lungs where oxygen passes into the blood. So it means that there's a lining. You can see there's a lot of O2. So, and this is the capillaries. So you want to know what is this layer? How did it penetrate into? So this is the alveoli. Part 2. State the site of filtration of blood that undergoes the ultrafiltration in the kidney, which is the glomerulus, not the Bowman capsule. The Bowman capsule receives the filtrate. Okay. Part 3. State the name of the process in which products of digestion move into the cell and are used to become part of it. So it becomes part of it. It's not absorption. It's like they will use the digester or the absorb and use it to make it to build like your muscles. So these are all called as assimilation. Part 4, state the name of the process in which excess amino acids are broken down in the liver to produce ammonia. So this is deamination. Okay, be careful. It's not animation. It's amination. A-M-I, not A-N. Okay, so be M first. And state the name of the organ that releases estrogen into the blood, which is the ovaries. Okay, so that's it for this paper. If there are any questions, of course, do feel free to ask in the comment section below. So thank you for watching this video. See you guys in the next one. Bye bye.